Are you diving into cell culture experiments and feeling overwhelmed by the sheer number of cells to choose from? You're not alone. Many researchers face the same challenge. But here's the good news. Selecting the right cells doesn't have to be a daunting task. In this video, we're going to simplify the process for you. We'll walk you through 6 key steps, beginning with defining your research objectives and then covering 5 essential questions to ask before making your selection, so you can start your research with confidence. Let's get started. The first step in selecting the right cell is to clearly define your research objectives. What are you trying to achieve with your experiment? This will guide your choice of cells, making sure they align with your study's goals. For example, if you're studying cancer, you might choose a cancer cell line that closely mimics the tumor you're studying. If you're testing drugs, you'll want a cell line that's responsive to the treatment conditions. Once you have your objectives in mind, it's important to consider whether a cell line or primary cells are the better fit for your research. This choice really depends on your specific needs. Cell lines are great if you need something consistent and easy to maintain, perfect for high-throughput screening, genetic manipulation, longitudinal studies, and other standardized conditions. But if you need a model that closely mimics the natural tissue environment, primary cells might be your best choice. They're commonly used in personalized medicine, disease modeling, and studies requiring cells directly from tissues. Now that you've nailed down your research objectives, the next step is to evaluate the key criteria for choosing the right cell line. To do that, here are 5 essential questions to ask. The first question is it authentic? Ensuring your cell line is authentic is crucial because using a misidentified or contaminated cell line can throw off your entire study. You need to verify that the cell line is exactly what it's supposed to be, since this directly impacts the reliability of your research. The go-to method for checking authenticity is short tandem repeat (STR) profiling. This technique creates a unique genetic fingerprint for the cell line, which you can then compare against reference profiles in established databases. However, it's important to note that STR profiling is primarily effective for human cell lines and some mouse cell lines. If you're working with non-human cells, you'll need to use species identification methods to prevent cross-contamination. It's a good habit to make regular authentication checks part of your lab routine, especially when you're working with new cell lines or after they've been in culture for a long time. Next, ask yourself, is it free of contamination? Contamination can quietly alter cell behavior and ruin your results. Whether it's mycoplasma, bacteria, fungi, or cross-contamination, keeping things clean is critical. Regular screening is a must, especially for mycoplasma, which can go unnoticed but cause big problems. Techniques like microscopy-based observation, PCR-based assays, DNA staining, or enzyme-based methods can help identify contamination. If you find mycoplasma, don't panic. Our anti-mycoplasma treatment reagent can eliminate it without harming your cells. Check the link below for more details, and combine this with strict aseptic techniques and regular checks for the best results. Now that we've covered the first 2 questions, the third question to ask is, does it exhibit the right biology? Ensuring your cell line exhibits the right biological characteristics is just as important as keeping it contamination-free. Your cell line's growth traits, like morphology, doubling time, and adherence, must align with your research goals. If it doesn't behave as expected, your results might not reflect real-world biology. To stay on track, regularly observe your cells under a microscope. Look for consistent morphology that matches what's described in the literature. Check that the doubling time falls within the expected range. Running a few preliminary experiments can also help confirm that the cell line responds correctly to stimuli like drugs or growth factors. This is essential for validating your research. Another crucial question is it genetically stable? Genetic stability is key for reproducible experiments. If your cell line undergoes genetic drift changes in its genetic makeup over time, it may not behave consistently, leading to unreliable data. To keep tabs on genetic stability, consider regular karyotyping or other genetic tests. These help you compare your cell line's genetic profile over time with the original or reference profile. If you notice significant genetic changes, it might be time to start a new culture from a lower passage number or even switch to a different cell line. Keeping an eye on passage numbers and avoiding excessive passaging can also help maintain stability. This brings us to the next question. Is it low passage? Low-passage cell lines are closer to their original state, meaning they're less likely to have undergone significant changes. High passage numbers, just like genetic drift, can lead to alterations that make the cells less representative of the original tissue or disease model. To ensure your cell line remains reliable, document the passage number of each subculture and regularly check these records. If your cell line is approaching a high passage number, consider reverting to an earlier frozen stock or acquiring a new low passage line to keep your experiments on track. As you can see, there are many factors to consider to ensure success in your cell culture experiments. But don't worry, we're here to help. We'll be sharing all the basics and the latest updates on everything cell culture through our videos and vlogs. If you have any questions, feel free to leave a comment or reach out for technical support. And don't forget to subscribe, like, and follow us for more content like this. Thanks for watching, and I'll see you in the next video.